And so we know that different viruses have different shapes, morphology, or sizes. But I mean, these differences are really just a manifestation of their genetic differences of their genetic materials. And all of that, the proteins, their shape, and how they infect are encoded in their genetic materials. It's kind of the blueprint. So if we can detect the genetic materials, not only we know that there's a virus, and we can know precisely what kind of virus is there. Um, so before we talk about detection, we need to refresh on the knowledge of the genetics. And so the genetic materials can take two forms, um, RNA or DNA. The difference is that the DNA lacks an oxygen atom on its backbone compared to the RNA. But this makes the DNA much more stable than an RNA molecule. But regardless, both RNA and DNA follows very simple base pairing rules, namely the G, the base C pairs with the base G, and the base A pairs with base T in DNA. And in the case of RNA, it is the base U pairs with base A. So these pairing rules form the basis of the genetics. And because of this pairing rule, so as long as there is one strand of genetic material, be it DNA or RNA, all other strands can be formed by following the base pairing rules. For example, if only this strand of single strand DNA is present, then by following the GC, GCC, GAT, AT pairing rules, the other strand can be formed. Um, and the other strand is usually called the complementary strand to the first strand. And so in a very similar way, and we can make RNA out of a DNA template strand, and a DNA similarly from RNA template strand. The viral genomes, or its complete genetic material, can be one of the 7 different forms, depending on whether it's made of DNA or it's made of RNA, and whether it's a single strand or double strand, and when it's single strand, whether it encodes the same sequence or the complementary sequence compared to the mRNA, which is RNA that is used to produce the viral proteins. So for example, the genomes of virus can be of the positive strand RNA, and in this case, its genome has the same sequence of the messenger RNA. But when the virus needs to reproduce itself, it has to be first to turn into a negative strand RNA, the complementary sequence of its genome, and then the complementary sequence can serve as a template to produce many, many copies of its own genome. And there's all other forms of the viral genomes, and we're not going to touch on today. And in the case of coronavirus, it is a positive sense, a single strand RNA virus. Its genome itself can be used to directly produce viral proteins, serve as a messenger RNA, or like we mentioned earlier, it can be used to generate negative sense, the complementary genome RNA, and which can be used as a template itself to regenerate the genome. And this process, the regeneration from the positive sense of a single-strand RNA genome to the negative sense of the RNA, is through the RNA-dependent RNA polymerase, which is the target of some drugs. And to sequence the viral genomes, we need to collect the virus, and we can get the virus from the BALF, the liquid that flushes the lower tract of the respiratory tract. Um, so we will then first confirm that there's a virus by using the test of the cytopathic effect assay. And once we confirm that there's virus, and then we can do the genetic material extraction. To construct its genome, we need to be able to sequence its entire genome. But the viral genome is usually much, much longer than any technology can sequence directly. So one need to take a different approach, a so-called shotgun approach or shotgun sequencing. So instead of sequence the entire genome at once, we can break the genome into small pieces. And critically, these small pieces has overlaps right there and here. And then based on these overlaps, they can then be back assembled into contigs and then assembled into the full genome. And each of the fragments here is small enough to be sequenced on a modern DNA sequencer. Of note, so within these fragments, there's also like DNA and RNAs from human cells and other microbes living in our respiratory tract. And so these sequences will be aligned to the known genomes and that need to be filtered before we can reassemble the unknown viral genome. The technology to sequence the fragmented DNA pieces is known as short-read sequencing or next-generation sequencing. Sometimes it is also called a second-generation sequencing. The way it works is via a chemistry called sequencing by synthesis. So basically, we have each fragment immobilized on a solid surface, and then a single nucleotide is added to this solution. And if the nucleotide is complementary to the position um, on the immobilized fragment, it will be added to this growing chain. And different bases, like A, C, T, G, is conjugated to a different color. So if you view from the top, and based on the color, we can then deduce what kind of nucleotide is added, and then we can know its complement basis um, on the fragment to be sequenced. And if we keep going for each position along this fragment, then we can get the full sequence of every fragment. A signal generated from a single molecule is too weak to be detected in practice. So in practical equipment, a single fragment is amplified to a cluster before it is being sequenced. So here I will introduce two ways that implemented in commercial solutions. One is implemented by Illumina. This method is called bridge PCR. We'll discuss PCR later, but here you just know that a single fragment, once immobilized on the surface, its complement strand and the complement of its complementary strand can be generated at nearby locations and to form a cluster of the fragments of the exact the same sequence. And alternatively, fragments can be embedded into a circular DNA. And then you can imagine a complementary DNA that goes around this circle and it generates many, many copies of the same circular DNA sequence. And this will form a cluster, and this is embedded later on to a solid surface to be sequenced. And so with the full genome sequence, one can compare it to all the sequences of known viruses and microbes. And this is how they found out that the COVID-19 is closely related to the SARS-1 that first emerged in 2003, and some of the coronaviruses carried by bats. And in addition, we can compare the different regions of sequence to find their similarity, and we can identify some regions of the very conserved sequences, which means that this sequence is similar across different coronaviruses, and such as this WRF1b gene, and this is RNA-dependent RNA polymerase, a very critical gene for the virus. Um, and so we can also use the known genes from the other coronaviruses and use their similarity to assign the tracks of the novel genome function. For example, this region is aligned to the S gene of a known virus, and this S gene, or called spike gene, um, is a gene that encodes the spike protein on the coronavirus surface, and this spike protein is responsible for the cellular docking and entry. Um, we can also compare the genome sequence from different locations, and this allows us to deduce how it spreads spatially over time. And so, for example, here, we can see that the East Asian and South Asian um, shares many of the COVID-19 strains, whereas the European and American shares a lot of the similarity in the strains and identified in these areas. And so very importantly, um, this comparison, this tracking of the evolution of virus allows us to see the mutations that are generated over time as the virus keep on spreading. And so we can know how fast it is mutating and in which gene it is mutating. And this is very important for drug design and for design reliable detection methods. And to detect the virus, of course, we don't have to sequence the genome every single time for every people. We only need to test the presence of the sequence that are mostly closely conserved in the COVID-19, that is has much less mutations, and it can distinguish itself from the closely related coronavirus like SARS and MERS. And so different countries have designed their own guidance for genetic detection based on this principle. And so, for example, the China CDC has advertised the use of a small fragment within the WRF1AB gene and the N gene and to test their presence in order to detect the virus. The technology to detect the presence of small piece of DNA is called reverse transcription polymerase chain reaction, or reverse transcription PCR, or RT-PCR. It needs this reverse transcription because, as we just said, the coronavirus is a single-strand RNA virus, and its genome needs to be first converted to the DNA. And so we use something called a primer, which is a short DNA sequence, and it can be chemically synthesized. It binds to the RNA genome, forming this base pairing with it, and then we use an enzyme called reverse transcriptase to make the DNA copy using the RNA genome as a template to form its complementary. And this DNA, because its sequence is complementary to the RNA genome, is usually referred to as the cDNA. And then the next step is to amplify the segments from the cDNA and test whether it contains the target sequence. Because usually one may only collect the tiny trace of the virus, and this DNA is so little that we can't really see it. So we have to amplify the DNA to a amount that's sufficiently large to visualize. And to amplify the DNA, we use a primer again, then bind to the region that we want to amplify. And once it binds to there, we use a DNA-dependent DNA polymerase, or just polymerase in short, to carry out the strand extension to form the new DNA. And now here comes the critical part. We have to separate these two strands for the primer to bind to it again. So heat up the DNA to 95 degrees to separate the template DNA and the newly formed DNA strand. And then we lower the temperature so that the primer, which is in much higher excess compared to the input DNA, it will compete and bind to the DNA template. And then we rise the temperature so that the extension can start. And so this is called a cycle. And in this cycle, the amount of DNA is doubled. If we keep going this cycle for 30 rounds or 35 rounds, then the DNA can be amplified for a billion times. And this cycling and this exponential amplification is the mechanism that makes the PCR extremely sensitive. The temperature change within a cycle needs to be done fast. And this can be achieved by something called the Petir effect, by injecting a high current through two different conductors, and results in a net heat transfer in either way, either heating or cooling. And so that needs a very special equipment called thermocycler to carry out. And this is the expensive piece of equipment. The PCR is not quantitative, and sometimes it is useful to know how much virus is present in the sample. And this is done by a method called quantitative PCR or qPCR. The trick here is to add a dye which can preferentially bind to double-strand DNA, and when it binds to double-strand DNA, it becomes fluorescent and can emit stronger fluorescence. And when there's only single strand DNA or single nucleotide, the dye is not that strong fluorescent. And so going through the cycle, we can add a stage where we measure the DNA concentration. So as the DNA is amplified through in cycles, the more cyber green dye will bond to the DNA. And so we can read out fluorescence at each cycle, and we can see the fluorescence will go up as the cycles get increased, and the fluorescence will keep going up. And the more input we start with, the earlier this fluorescent signal will pass a preset threshold. And the lower the input, the later it will pass. At later cycles, it will pass this preset threshold. And so we'll keep track of the cycle at which the fluorescence signals pass the threshold, and we call this cycle number a CT value. And so this CT value is a very nice indicator for or directly proportional to the amount of input material. And so using CT values, then we can have a quantitative measure of the input material. Because qPCR needs the fluorescence measurements, it needs much more expensive equipment for that purpose. So we know that the primer dimer is of different length and sequence composition compared to the target sequence. And so what we can do is we can heat up the entire reaction mix after the amplification is finished. And we know that the DNA will be separated at higher temperature. And when they get separated, we shall see a separate green signal drop because it will be unbound from the DNA. And then we can measure the fluorescence signal at different temperatures. Because the primer dimer is shorter, and so it will be first separated or be separated at lower temperature. And if we take the derivative of the fluorescent signal, then we should see a signal corresponds to the primary dimer. And the target sequence is longer, and so it will be separated at higher temperature. And if we take this derivative, and we should see a signal corresponds to the target sequences. And so by doing this analysis, which is also called the melting curve analysis, we can then find out whether or not we have the target sequence in our sample. And sometimes you get the signal passing a CT value, and there's no target sequence. There's just purely primary dimers. So this gave us a way to reduce those false positives. We often heard that nucleic acid testing has false negatives in addition to false positives. And those false negatives are probably even worse for the purpose of detecting active viral carriers. But in fact, the PCR itself is very sensitive. It can detect as little as a single DNA molecule. But there are other factors that often limit the detection sensitivity. For example, the viral load. So this graph shows that the upper respiratory tract has much lower expression level of the ACE2 gene compared to the lower ones. And we know the disease mainly infects lungs and this lower respiratory tract. But we know that we primarily sample the upper tract, and we get much lower load because of this. And during sampling, sometimes we can even miss the target or get very little viral samples if the sample is not very proficient at doing the sampling. And so we can get the sampling error. Besides, the sample has to be shipped to a specialized PCR laboratory for testing, and during the transportation, the losses can also occur. And because of all of these factors, it is estimated that one may have a false negative rate of 30% for the nucleic acid testing. The solution is to do multiple tests to reduce this false negative. For example, if the false negative is capped at 30%, then do it 4 times would effectively make this test 99% accurate.